So everybody wants to know what works, right? We have a little saying at the Dry Eye Foundation, or maybe it's just me that just keeps saying this thing: everything works for someone and nothing works for everyone. How do we find our way to what works for us? That's been kind of the subject of a lot of the discussion today. As you've seen, a lot of what we're going for today is different angles on things, coming at something from kind of a fresh, new view. So we're going to look at what works in a few different ways. First, we've got Dr. Brown and Dr. Jacobs, and then this is session 4. Then in session 6, we're going to have the patient speakers come back, and we will be wrestling through what works from a patient standpoint, not just medically, but with all of the other aspects of the struggle that we've talked about. So take it away, Dr. Brown.
All right. I'm Dr. Sandra Brown. I'm the medical advisor for the Dry Eye Foundation, and I'm going to talk to you today about optimism versus evidence, data limitations and marketing hype in dry eye treatment. And I kind of made a few little rumblings about that earlier during the panelist questions, but we're going to dive into it in more detail. But I want to start off by saying that I really hope that my lecture makes you feel better about treatments that didn't help you. Because I know that a lot of times you go from treatment to treatment to treatment, and you feel as though you are potentially the only person for whom multiple treatments are failing. You might be thinking, what is it about me? Is my dry eye more mysterious, more severe, more intractable than everyone else's? All of these fancy medications, all these expensive medications are not helping me. What is wrong with me? I'm going to tell you what's wrong with these medications. In other words, I'm going to keep you awake after lunch by disrespecting industry. So you say, well, will this amazing new treatment work for me? We're going to talk about optimism versus evidence. Now, what's optimism? It's what pharma tells your eye care provider, what key opinion leaders tell your eye care provider, and what pharma tells you. And then also what your eye care provider tells you. But what they're telling you is what they've been told by these other entities with a vested interest. How about evidence? Evidence is what the FDA clinical trials show. And that can sometimes be very, very different. I want to start with a very important sidebar. It's a sidebar, but really it's a central bar. When you listen to the rest of this talk, and also when you think about this for yourself, you have to define your own definition of work. Does this treatment work for me? What is your goal? Your goal for this treatment, okay? I feel like your goal should be that the treatment makes you happy. How can it make you happy? It can decrease your symptoms, increase your function, decrease your treatment burden in terms of time or money. You can pick one or all of those as an acceptable definition of work for me. Now, there's another definition, which is that the treatment makes your doctor happy. Well, how does your doctor get happy? Your doctor gets happy because your examination is improved. Now, if your doctor really cares about you and you come in and your exam is better and you say you're feeling better, then your doctor is simultaneously ecstatic and relieved because finally they're making progress. But making your doctor happy should not be your definition of something that's working for you. Okay, let's take a couple slides on how FDA clinical trials are designed. First of all, they're designed with the structure of drug versus placebo. All right, who in this room knows what a placebo is? Okay, so I got the notion that a lot of you have read a lot on your own behalf, which is awesome. Okay, the important thing to understand is that when you're studying an eye drop, you're studying a drug molecule. So you're not studying the whole eye drop, you're studying a specific molecule or a combination of molecules in that eye drop. And so the placebo is going to be everything else except the molecule. All right, and the assumption is that the placebo is completely inert, doesn't do anything to you, good or bad. A synonym for a placebo eye drop is vehicle, so I might kind of casually use those terms interchangeably. They mean the same thing. The goal of a placebo eye drop, which is more difficult to make than a placebo pill, is that it has to be as similar as possible in all respects to the actual drug being tested, and ideally it would have the same patient experience. So let's say that the drug being tested stings when you put it in. Well, ideally, the placebo would sting when you put it in. Because what happens if you have two clinical trial subjects in the same waiting room and one of them says, this stuff stings twice a day for 5 minutes, and the person sitting next to them is like, it's never stung me and I'm on my second bottle. Well, if that happens a couple of times, it's possible that a patient may figure it out or make a decision on their own behalf. It's not stinging, it must be the placebo, okay? So it can be challenging to make equally stingy or not stingy eye drops, but that would be, in my ideal world, the goal of a well-designed placebo eye drop. But what if a placebo eye drop isn't inert? What if it does something? It could do something good. It could do something bad. Could a drug sponsor, which is the company that's manufacturing the drug and trying to get it approved, could they manipulate their placebo? Color me paranoid, but we'll talk about that in a few slides. Let's also talk about clinical trial participants. The clinical trial participants are all little white airplanes flying to the east. These people need to be as similar to each other as possible so that there's no extraneous factors that are affecting their response to the treatment. Okay, drug gets approved. It's approved for little white airplanes flying east. And if you read the drug label, you'll see that. You'll see that it was tested on a specific age range. You might be younger, you might be older. If you dig a little deeper into it, you might read a long list of conditions that the trial subjects did not have, and you might have one of those conditions. So you might be a little orange airplane flying in a different direction. This means you are not like those clinical trial subjects. This is typical post-approval prescribing, by the way. So a drug company needs to get their drug approved. They can get it approved for a patient population which represents 0.0001% of people who live in Kansas. They don't care, because the minute it's approved, it's going to be post-approval off-label prescribed like mad. And that's where the billions of dollars are. All right. Now, let's get into some actual clinical examples. How many people have been prescribed Xdemvy, just out of curiosity? Okay, so, some, not all. All right. Well, Optimism, Xdemvy. Evidence? Did you know this? There's no symptom data in the Xdemvy clinical trials. What does it do? It kills Demodex mites that live on your eyelid margins. And Demodex mites are one cause of blepharitis, not the only cause. That right there in the middle is a picture of a Demodex mite hanging on to an eyelash, and the eyelash has been yanked out of some unsuspecting person's eyelid. So this is one way you know for sure a patient has Demodex, is if you actually take eyelash samples. You can't just reach up and pull them out. You have to kind of twirl them around first and then pull them out. That's in the clinical trial publication. So do you think we do that in the office? How many people who've been treated with Xdemvy have also had their eyelashes pulled out? To check. Okay, to check you for Demodex? Okay, I should specify it, to check you for Demodex. So we're zero. Right, okay, so zero. All right. Well, so how do we decide you might have Demodex? Well, there's a particular clinical sign called eyelash collarettes, and you can see that in the picture below on the left. It's basically Demodex poop. And if you have that, then that is associated with the possibility of Demodex infestation. And this is Xdemvy in action on the left, eyelash debris on the right, no eyelash debris. So that's great, right? You want that, all right? What do we know Xdemvy does? We know it kills Demodex mites and improves blepharitis. So who's happy? Your doctor's happy, because they look at you with a slit lamp and they say, hey, your blepharitis is better. There's a whole lot of optimism between killing Demodex mites and you being happy. What has to happen? Your blepharitis is better, your meibomian gland function improves, your lipid layer of your tear film gets thicker, slows your tear evaporation, improves your tear osmolarity, and ultimately, at the bottom of all those arrows, it decreases your symptoms. And that's what you want. That would make you happy. Well, actual evidence for symptom improvement is none. So there were 2 randomized clinical trials involving 833 patients, of which over 400 received Xdemvy, and there was no symptom data collected in the clinical trials. Don't believe me? That is a screenshot from Daily Med. The indication for Xdemvy is for the treatment of Demodex blepharitis. And there's not a word in there about improvement of symptoms of dry eye.
I was recommended for Chalizion.
We'll call that off-label. Okay. So who's happy? No questions during the talk, please. Your doctor's happy if your blepharitis looks better. You are not happy if you don't feel better. So please take home this message. XDEMVY was an ineffective treatment for you. And above all, please do not repeat it. If it didn't work the first time, it's not going to work the second time. All right, here's why you should feel better that XDEMVY didn't help you. Well, number one, you didn't have Demodex blepharitis to begin with, and XDEMVY only treats this one highly specific condition because it doesn't really treat general blepharitis. It kills cooties, kills Demodex. Number two, you're in good company. There's no evidence that XDEMVY actually improves ocular surface symptoms, even in patients who had proven Demodex blepharitis, okay? I'm not saying that it does not improve their symptoms. I'm saying that the drug sponsor, for some reason, elected not to include a symptom measure in their clinical trial, which puzzles me, all right? But that's XDEMVY. That's one example of how a drug is approved for an indication that then has many, many arrows of assumption to actually get to the point where you believe that this medication is going to actually make you feel better. In other words, you're happy. Okay. All right. Next, a little bit of a different game we're playing here. Optimism, Restasis, evidence, skip the cyclosporin. And my colleague actually kind of created a nice intro to this conversation to talk to you about Restasis. This is amazing. So Restasis was actually approved when I had been in practice for a couple of years, and I remember the drug reps coming around and pushing it. And this was back when drug reps could buy you steaks, fly you to Hawaii, and I mean, they were in our faces continuously. So the original Restasis trial was quite sensibly designed with symptom reduction as the primary outcome measure, right? That's what you want. You want an eye drop that treats ocular surface symptoms that are presumed, back then, okay, presumed to be due to ocular surface dryness. Okay, that approval was denied. Why? Because there was no difference in the symptom measure outcome between the drug and the placebo. What does that mean? Does it mean that neither one made anybody feel better? No. Remember, this is a competition between a molecule, which is cyclosporin, in an eye drop, and then everything in that eye drop except the cyclosporin. So what happened? Well, cyclosporin didn't win. And the FDA said, no, we're not going to approve your drug, which is cyclosporin emulsion, because your emulsion seems to work great. Go sell the emulsion. This is the vintage data. This is a scanned document. You can't find an electronic version of this document. It says, per the FDA statistician, no significant among group differences in symptoms of dry eye or treatment success. But somehow that translated into billions of dollars. Well, how did they do that? Well, Allergan had to try again. They had to get this drug approved somehow, and this is why I call Allergan the original bad daddy of small font evidence, all right? They tortured their data, won't go into all the torturing, but they eventually got it improved for an indication of increasing tear production. Okay, who's happy? Your doctor, if your Schirmer increases. Are you happy? Maybe, maybe not. You paid how much for this? So remember all those little white airplanes? So not only did Allergan have to give up on the symptom measure altogether, but the way they tortured their data was that they narrowed their subject population to patients with moderate to severe keratoconjunctivitis sicca, okay, so the older term, due to inflammatory problems, okay? So if you are not somebody who has an inflammatory version of moderate to severe keratoconjunctivitis sicca, you are an orange airplane. Well, suppose you're a white airplane. How often is it the case that Restasis is going to help you? About 15%. 1.5, not 5.1. So if you exactly resemble all those white airplanes, you have a 15% chance of having an increase in your Schirmer score. So reasons Restasis or cyclosporin as a drug in general didn't make you feel better. You weren't a white airplane. You didn't have an inflammatory cause for underproduction of aqueous tears. But also you're in good company again, because 85% of clinical trial subjects saw no benefit. So I hope you're getting a little bit exasperated with industry. This is where I live. That's my baseline. So somebody says, you need to be on cyclosporin. In the old days, we would say, try this instead, Refresh Endura. Second funny fact was that Allergan said, oh, we got a great dry eye drop here. Let's sell Refresh Endura. And then the ophthalmology community kind of twigged to that. And also we were really mad about how much Restasis cost. And we were telling people, remember I was there, I lived this, telling people, did you live this too? Go out and buy Refresh Endura. What do you think Allergan did? Yes, took it off the market. Okay. So the closest thing you can get now is Refresh Digital PF. So if someone says, I want you to try a cyclosporin agent, you can say, pause momentarily while I go try Refresh Digital PF. Well, so what happened? Allergan invents the beneficial placebo eye drop. They did not do it on purpose. But let me tell you, industry, everyone else said, we are never making that mistake. Okay? Because Allergan had to fight hard to get Restasis approved. And now, you know, they're not the only people playing on the field, which they were for decades. So anybody coming along now has competition. So they cannot risk sinking a huge amount of money into a drug, which is not then a blockbuster drug. So let's talk about MIBO. Optimism, MIBO, right? And there's a lot of concern about meibomian glands these days, thus MIBO, spelled with the I and the E backwards, by the way. And so there would certainly be a lot of interest in a medication that we don't know how it works. But whisper decreases the rate of tear evaporation. Let's talk about that clinical trial. So we talk about placebo. The placebo was a back-preserved hypotonic saline. And in the clinical trial documents, which I've read, there was no mention that this was buffered. How many people here are sensitive to back as a preservative? Yep, okay, so hands go up, all right. And how many people here know what a buffer does? Lots of people know.
Are you saying buffer?
Yes, buffer. Can you hear me now? Yeah. Okay, buffer, all right. So a buffer maintains a liquid at a certain stable pH. So if you have a back-preserved saline as your placebo, it can make symptoms worse in back-sensitive study participants. And if it's not buffered, the pH is going to drift acidic, and that'll make symptoms worse. And so this is where I'm scratching my head hard, / pulling out my hair, because I cannot see any reason why they didn't use a preservative-free buffered hypotonic saline in identical packaging. They could put both these products in multi-dose preservative-free bottles, and nobody would have any idea which one they were getting, and that would have been a fair placebo. In fact, the placebo that they used was so overtly unfair that, okay, ... right? Maybe it was calculated. I'm speaking on behalf of myself at the moment, I should say, not necessarily the Dry Eye Foundation, but this really aggravates me. Okay, optimism, MIBO, evidence, lies, damned lies, and statistics. Who in this room is old enough to know that Mark Twain coined that lies, damned lies, and statistics? Okay, I'm old. All right. Well, MIBO is approved for treatment of signs and symptoms of dry eye disease. That's the FDA indication right down there. Okay, whoopee, we've got symptoms. And it's marketed, amazingly, as directly targets too much tear evaporation. The drug label says nobody has any idea how it works. It's hypothetical. Okay, well, I guess they got a little bit of a smackdown on that, because now it says protects against tear evaporation, but at the bottom, dramatization. The exact way MIBO works is not known. Okay, I have never in my professional career seen a drug marketing dramatization. I don't think that's legal, but okay. Let's talk about how symptoms were scored in the clinical trial. They were scored with the classic visual analog scale. Stick with me through the science, okay? I'm trying to keep it engaging, and I really think you're going to understand at the end. All right. So visual analog scale is a scale from 0 to 100, and you say, okay, my pain today is a 4. No, it's never a 4. My pain today is a 44. It's an 84. It's a 94. Using this visual analog scale, you then compare the patient to themselves. So before they started treatment and after they started treatment. This is important that you can only compare the patient to themselves because my 47 is probably not your 47. Okay. All right. And because a lot of things can affect how you will score your pain on a particular day, your mood, what you had for breakfast, whether the barista in Starbucks got your drink just right, whether it's a nice humid day outside, a lot of other kind of external variables can affect your VAS on a particular day. Most studies will define a change in that score that they consider to be significant, meaning that it's actually due to the medication. So it's pretty typical to define this as 30% or more from baseline. So if your VAS was 100 and it went to 70, you're a responder. If your VAS was 30 and you went to 21, you are a responder. Now let's talk about how this data was advertised separately from the dramatization. This is what was shown to providers. I saw this all over the place when MIBO was first approved. What you see is, day 0, day 15, day 57. And you see this gap between the gray, which was the placebo, and the purple, which was MIBO. Who knows what a p-value is by any chance? Because you all are like super educated, smart people, and it's so fabulous to have all of you in this room. So you know that the more zeros there are in a p-value, sort of the better it is, the stronger it is, the more statistically significant the effect is. Okay, there's a lot of zeros in that p-value there on the right-hand side, but look at the actual difference in the numbers, right? From an average VAS score of 28.5 to an average VAS score of 19.3. So that's, 9-point decrease. So that's the average population, and they got an average of a 9-point decrease. So a question that I asked immediately, which I've not gotten any rep to answer, is, do you classify that as a meaningful to an individual patient decrease in their VAS? And what's inside that? Are there patients that got a whole lot worse and patients that got a whole lot better? Did everybody get a little bit better? And they didn't provide that information at all. Plus, how much better would that control score have been if they'd been using a fair placebo? Now this just confused the living daylights out of me. Big purple blob, 43% improvement from baseline in eye dryness at day 57. Same question, did everybody get 43% better? Did 43% of the patients get 100% better? I mean, this is just confusing, but it looks great in an ad. So I'm going to say that the MIBO folks are the new bad daddy of small font data reporting. So stick with me now, okay, because this is where we really get to the heart of how much this medication helps a patient, or how likely it is to help a patient, not a population of patients. You got to read the actual clinical trial, and you got to read the other secondary endpoints. Okay, healthcare providers in the room, how many people read the small font in the results section? Okay, all right. We are in the minority. Got a couple hundred of us in a room, and probably the same hands would go up. All right, don't worry about reading that. So remember, we have this 30% improvement or decrease in VAS score defines you as a responder. 57% of the MIBO subjects were classified as responders, and 47% of the saline study subjects were classified as responders. So, number one, 57%, which is not 100% of the study subjects using MIBO, had a beneficial effect. We don't really know the magnitude, okay? And number 4, in my opinion, unfair saline control study subjects, 47% of them had a similar effect. So that's a difference of 10%. How much is 10% worth? It's a 10% statistical probability that MIBO will help you more than saline. $800 a month. And I have to enjoy the 43% irony because we've got this big purple blob. Wow, on average, patients got 43% better. I want this for my patients, versus 43% of the clinical trial subjects did not notice a symptom reduction while using MIBO. I had to point that out. Reasons MIBO didn't make you feel better. Number one, excessive evaporation is not the primary source of your symptoms, even if you have so-called meibomian gland disease. Number two, you're in good company, because 43% of clinical trial subjects were classified as symptom non-responders to MIBO. So how many people here have been prescribed MIBO? I should have asked that in the beginning. Wow, a lot of people. Okay. All right. And my cornea colleague tells me that the first bottle can be had for a fairly decent price, and then after that it's an unbelievable amount of money. So if MIBO isn't making you feel better, more MIBO is probably not going to make you feel better. And you shouldn't feel hopeless. This should not add to your sense of hopelessness about your situation, that here's another medication, a medication that works differently from all the other medications that are on the market that's not helping you, because it didn't help a lot of people. All right. Drug clinical trial take-home points. Biggest one. Big Pharma develops drugs to get rich, not to save the world. They are past masters at promoting drugs to providers through advertising and the use of key opinion leaders, talking heads, okay? And most providers are way too busy to take that deep dive into clinical trial publications, in particular the small font under secondary endpoints. And that's typically the only place where those not so stellar outcome data can be found. Also, frankly, medical statistics confuses us too. So what can you do when your doctor recommends a new dry eye drug? First, if you want to try it, do not change anything else about your treatment regimen while trying a new prescription eye drop, especially if it potentially is going to cost you a fortune. If you're going to have to rent out your house and live over your garage to afford this medication, you need to take all the structural steps you can to be sure that this medication is actually substantially benefiting you. And that means you can't be doing anything different at all while you're trialing it. Once again, this is my opinion, but in particular, I do not think you should be undergoing in-office treatments while you're trying a new medication, because those can introduce a huge amount of confusion into what's working. You can end up in a really kind of financially scary situation where you feel like you need both the IPL and the MIBO. Can you afford that for life? Is it causing huge amount of stress and strain on all other aspects of your life? Potentially. Okay. Remember, your goal is to have a lower treatment burden, not to just add something that you're not sure works on top of something else that you're not sure works. I hope this is resonating. Okay. Circling back to that first slide about the sidebar on the definition of work, once again, for a new medication, a new prescription medication, you need to establish your own definition of this is working. And it can be any definition you want. Like you can pet your dog for 5 minutes, whatever. Your definition that it's working, not your doctor's, okay? And very, very important, you need to establish your own treatment duration, at which point you will decide whether the eye drop does or doesn't work for you, and you've got to stick to that duration. And I'm really kind of reflecting back on the Restasis game, where patients were kept on Restasis and we're told, well, it's not working at 6 months, it might work at 12 months. Well, it's not working at 12 months, it might work at 18 months. And they were on very prolonged courses of treatment to no avail. So I really want you all not to fall into that same trap with these new so-called novel, amazing, love the purple blob advertising prescription dry eye medications. So I have a little section on biologic eye drops, but I don't want to cut into Dr. Jacobson's time. How are we for time? Keep going? Okay. All right, so, Rebecca mentioned earlier that the Dry Eye Foundation really ramped up its patient safety, eye drop safety efforts a couple of years ago. I got asked a really astute question, was this in response to EsriCare? We actually started our eye drop safety campaign in the biologic eye drop space. That caught our attention before EsriCare ever happened, so I'm going to spend a little bit of time on that. Optimism, biologic eye drops, evidence, science geeks win. All right, first of all, there are a couple biologic eye drops that are not bogus. Your own blood products, which would be autologous serum tears, platelet-rich plasma, when properly prepared and stored, are not bogus. Oxervate is an FDA-approved single protein eye drop, and it's not bogus as long as you suffer from the condition that it treats, okay? It's what's called a recombinant protein, which means that it is made for us by specially trained bacteria. Why does anybody think they want a biologic eye drop? Providers believe that these products contain proteins, lots of different proteins, delicious proteins that are going to help the ocular surface to heal, help the corneal nerves to normalize and quit firing off all these random horrible signals, decrease inflammation, and just kind of do good stuff. Patients generally are looking for something different than all the prescription drugs that have failed them. Natural, preservative-free, biologic, not chemical. And, kind of the biggest pitch, which is that these products work with your body to heal itself. And the more that standard Western medical care has been failing people, the more that they are intrigued by this kind of offer, so to speak. This is why all other biologic eye drops are bogus. The whole premise of biologic eye drops is that they contain proteins, and these proteins signal cells, nerves, and all kinds of other structural and functional parts of the surface of your eye to become more healthy. But proteins fall apart. They become non-functional very rapidly under the following unpleasant circumstances for them. At room temperature, if not kept in a perfectly pH-neutral buffered solution, if not kept in a solution meaning liquid, with the right oncotic pressure, okay, don't worry about that, but I'm just throwing it in there. And then if you mix it with something that doesn't exist in the human bloodstream, like glycerin. So if you look at actual scientists using proteins for real science, they keep those proteins as a freeze-dried powder, they dilute them in liquid, and use them within minutes to hours. So if real scientists doing real science want to study a protein, the effect of a protein, and that's how they have to do it, why on earth would anybody imagine that you could have a liquid that's sitting on your countertop for days that contain functional proteins? Here's the answer from your typical biologic eye drop company. We have a protein analysis, all right? And I've read them. They're most often done by a company called Ray Biotech. I have nothing against Ray Biotech. They'll analyze anybody's proteins. You can send them your dog's water bowl, and they'll analyze that for saliva proteins. Okay. They will do what's called a protein analysis, and they'll get back a document. They'll send it to the company, and it'll have lists of hundreds of proteins on them with all these little concentrations on them. Hang on. Stay with me. Why proteins are not like celery. On the left, celery. On the right, a protein. This is one way to model the shape of a protein, and you see that it is twisted and folded. Proteins need to be twisted and folded in order to function properly. And if they untwist, unfold, or break up into little pieces, they are not going to work, okay? Well, what about that protein analysis? Chopped up celery tastes like celery. Chopped up proteins don't work. What do those lab tests identify at Ray Biotech? They identify unique fragments of proteins. So if there's a protein fragment in that liquid, they'll say, there's evidence that there used to be this protein there. That protein analysis from Ray Biotech or someplace similar doesn't say anything about whether the protein is intact and doesn't say anything about whether the protein is functional. In fact, when I called up their PhD person on the phone and said, so it's like this, she was completely confused. She had no comprehension of why I was asking her about any kind of liquid containing proteins that was kept out on a countertop at room temperature. because that's just not how it works. So the lab detects a celery fragment, and the eye drop company says, the lab says our product contains whole celery stalks, but that's not true. I wrote this very geeky article on this subject in 2009. We're going to skip that. Okay, so what have we learned about biologic eye drops? The companies that manufacture and promote them will use intense jargon and complicated but meaningless laboratory reports to convince providers and patients that their eye drops do something biologic related to protein activity. But they don't. So some of you, maybe many of you are thinking, why did my eye care provider recommend them? Okay, touchy subject, something Rebecca and I talk about a lot. So the most straightforward explanation is that healthcare providers are not accustomed to being flagrantly lied to by drug manufacturers. And we hate medical statistics, most of us. And anything we learned about proteins was somewhere between 5 and 25 years ago. And so we believe these people. And we have patients in a really tough situation, and we want to help those patients. And this seems like something different that could help those patients. And so if any of you have been encouraged to purchase a externally manufactured biologic eye drop by your eye care provider, don't be mad at them. They wanted to try anything and wanted you to try anything that could potentially help. And they just did not disbelieve the manufacturer. What can you do if your doctor should recommend a biologic eye drop? Politely say no. Remember, no does not include autologous blood products or oxyrvate. With this caveat, that oxyrvate has a very specific indication for the treatment of neurotrophic keratitis. If you do not definitely have neurotrophic keratitis, don't agree to use oxyrvate unless you have a conversation with your provider to say, I know it's being used for this indication, I don't have that. Why do you think it'll work? How long do you think I'm going to have to use it? And how much is it going to cost? Next time, devices.